Good morning. Welcome to Grand Round on this cold morning. Today it is with great pleasure that I'm welcoming a longtime friend of mine, distinguished researcher and colleague, Dr. ARemaley. Alan is trained as a clinical pathologist, as one of us. He obtained his MD and PhD from the University of Pittsburgh. That was followed by a clinical pathology fellowship at UPenn. And following UPenn, he went basically not too far away to NIH. And there he did his clinical laboratory service responsibilities as director of laboratories. But more importantly, he spent a lot of his time doing lipid and lipoprotein-related research. And in 2007, Alan became the section head of the section on lipoprotein metabolism of NHLBI. Now, this is not just any division or section. This is a really storied division. I think Alan's going to touch upon that because pretty much all the who's who, if you're in lipid and lipoprotein research, have worked there one time or another. And if you haven't, and if you studied any kind of lipids when you were in medical school or clinical chemistry, you will recall names like Fredrickson of the Fredrickson classification, Freewell of the Freewell formula, etc., etc., etc. So this is, of course, a great honor to be the chief of that particular section. Alan, of course, has received many honors and awards, way too many to mention. He's also on the editorial board of many distinguished journals. I'll just mention some of those that are my favorite journals, such as Arteriosclerosis, Vascular Biology, Journal of Lipid Research, Journal of Clinical Lipidology, as you get the idea, atherosclerosis, clinical chemistry, etc., etc. So I was very fortunate to make Alan's acquaintance at a time, probably 15, 20 years ago, I can't recall. When we were both active, we actually were successively chairs of the division of lipoprotein and vascular disease of the American Association of Clinical Chemistry. Now, with Alan's vast amount of expertise in this area, he has been my consultant whenever I have questions with regard to this fast evolving area of research. So without further ado, I would like to welcome Alan back to Minneapolis, and today he'll share with us his hot new findings, his exciting research on the application, the use of ApoC2 mimetic peptides in the treatment of hypertriglyceridemia. Alan.
Well, can everyone hear me? So I'd like to thank Mike for that kind introduction. And this is actually my third visit here. And as you heard, I used to think Mike and I were friends, but I noticed that he always invites me in the wintertime. And although this time there's less snow than last time, but I'm just kidding. I always love coming here. And as Mike said, I'm trained as a clinical pathologist, so I try to have a little bit of something for everyone, because I remember attending these kind of meetings, and someone studies genetics and biochemistry, so I try to put a little bit of everything in here, so I hope you enjoy it. And so I'm going to talk about ApoC2. So this is where I'm from. It's about 10 kilometers from downtown DC. So this is the famous kind of Building 10. This is the replacement. So it's a small hospital, about 300 beds. But it represents about half the research beds in the country. So everyone who comes here is on a clinical trial. And our lab and many labs in NIH are very interested in rare genetic diseases. So I'm interested in rare diseases related to lipid metabolism. So this project actually started from a patient with ApoC2 deficiency. So we have rounds each week, and we always think, what could we do for our patients? So this actually started from thinking about how we can help our patients. And as Mike said, I have two jobs in the clinical lab. I only get paid once, and then I have a job in heart, lung, and blood where I do research. So one might ask, why do we need new drugs? for cardiovascular disease where we have statins for lowering LDL. And as you all know, we now have PCSK9 inhibitors that lower LDL almost all the way. But you'll see, despite the fact that statins are wonderful drugs, that they aren't really the complete solution. So I'd like to start with a case. So this was a 55-year-old white male with a positive family history of cardiovascular disease, slightly overweight, slight increased blood pressure. He was a runner, so he had a very good exercise tolerance. And as you can see, his LDL was slightly elevated at 177. And he was started on Zocor. But like a lot of patients who take statins, they feel fine, and about half people discontinue after about 5 years. So this was true in this case. He discontinued after 3 years when he lost some weight and his LDL went down, and he thought he was doing fine. And then he went on to develop resting angina and had to get a triple bypass surgery. Now, this is a patient you all know. This is President Bill Clinton. And he had the best of medical care and the best of advice. And despite that, the statins have really failed him. And unfortunately, he's in good company in that all the presidents of the past century have died of heart disease, except for Ronald Reagan and Kennedy of some sort. So statins are very important drugs. But what a lot of people don't realize is that under the best of circumstances, that is well-controlled clinical trials, they only reduce events by about 30%. And in practice, we probably don't do as well as that. We can lower LDL now better with PCSK9 inhibitors, but it's not clear that from the early studies that will translate into a further reduction in events. And so there's been several early studies showing that if you combine statins with other drugs that modulate triglycerides or modulate HDL, that you can maybe further reduce cardiovascular events. So that was, this is old data, and this generated a lot of interest in drugs that are raising HDL, but that hasn't worked out so well. But niacin also lowers triglycerides. So that's what I'll be talking about today. So I'll start out with a brief review of the biochemistry of triglyceride metabolism. I'll talk about some clinical findings of patients with high triglycerides, talk about the current therapy, and then the work that I'm involved with in trying to turn ApoC2 into a drug. So these are the major pathways that keep Mike and myself busy for many years. So there's 3 cyclical pathways. You have the so-called exogenous endogenous pathway, and the main reason we have lipoproteins is not really to move cholesterol around, but is to deliver a triglyceride for energy metabolism. And the exogenous pathway, you have dietary lipid going in. It's packaged in chylomicrons. These go out to the periphery, and I'll go in more detail, but they undergo lipolysis. They release the triglyceride, as I said, for energy metabolism, and then they turn into remnants, and then they're taken back to the liver. The liver takes in free fatty acids as well as dietary triglycerides, resecretes it as VLDL, particularly in fasting. This also undergoes lipolysis, and some of the VLDL is converted to LDL, and that also goes back. And the other major pathway that there's great interest is the HDL, so-called good cholesterol. And it's thought to be beneficial largely because it mediates the removal of excess cholesterol. But I'm not going to really talk too much about that today. So Mike mentioned I'm at the Laboratory of Lipoprotein Research, and this is my scientific grandfather, Don Fredrickson, who unfortunately passed away about 10 years ago. And ApoC2 deficiency in many of the diseases I study, it was first described by Don Fredrickson. But his legacy, and this will be important for my talk as we go on, is that he trained really wonderful people, many of which unfortunately have passed away or have retired. But there's still a few people that some of you may recognize, such as Tony Gatto and Virgil Brown, who are very active, and I'll talk about Virgil later on in my talk. Also a little about history. He's not in this picture, but Ed Korn, who just retired after 65 years of research at NIH. He discovered in Don Fredrickson's lab lipoprotein lipase. And the other one I'm going to talk about today is ApoC2. So ApoC2 is a small protein activator of LPL, and you can see that was discovered by Don Fredrickson and Virgil Brown, and this is a current picture of Virgil. So this is the endogenous pathway. As I said, there's 2 pathways for triglycerides, the exogenous and endogenous. And endogenous is the VLDL. The 2 pathways are very similar, but the endogenous pathway starts when fatty acids or triglycerides are packaged in the core of VLDL, and you have these various proteins on the surface that modulate where the lipoproteins go. These eventually go out to the periphery, and then you have this key enzyme, LPL, which hydrolyzes triglycerides. ApoC2 is an obligate cofactor in the lipolysis, and this is converted to remnants. So most of these remnants go back to the liver by after interaction with proteoglycans or various ApoB or E related receptors. Now, about maybe half the VLDL undergoes further rounds of lipolysis and then converted all the way down to LDL. So LDL is a triglyceride-poor, cholesterol ester rich particle, and as you know, this is what causes all the problems because you have too much LDL. Some of it infiltrates into the aorta where it causes atherosclerosis, and then the rest, of course, goes back to liver by the LDL receptor. So the reason, as I said earlier, we have LPL is for energy. So the LPL is actually produced by the underlying tissue, but it has to get out to the surface because that's where the lipoproteins are. So in this case, you have a lot of LPLs made by skeletal muscle. It's transported as a very complex trafficking pathway, and it goes out to the surface, and it's docked on what's called the GPI-HBP1 protein. There's an ionic interaction, and it waits for lipoproteins to come by. So you have ApoC2. C2 comes in, and there's a physical interaction you'll see between LPL and C2, activates the enzyme. Fatty acids are generated, and they're very efficiently sucked into the tissues. They don't usually leak out into the plasma very well, and they go into the tissue for energy metabolism. This dissociates, and then HDL is the reservoir. So when C2 or the VLDL is consumed, it jumps on HDL and it waits for more chylomicrons to come out, and you repeat the cycle. So as you can imagine, this process has lots of need for regulations. So depending on whether it's in the fed or fasted state, you want to send energy, you want to burn energy in your skeletal muscle, or you want to store it. So you have proteins that turn off and on very quickly LPL. And some of the proteins that are very important is this chaperone protein, lipase maturation factor that brings it out to the surface. I mentioned GPI-HBP1. ApoC2, which I'll talk about more, is the activator. And then you have ApoA5, another activator. And then you have very important inhibitors that antagonize this path, such as ApoC3, angiotensins, which are newly described proteins that turn off LPL, and then ApoC1. Now, there's not a lot of human data on ApoC1, but the data from transgenic animals is very convincing that human ApoC1 is probably a very important negative regulator like ApoC3 of this pathway. Now, this is the best model for how we think LPL activates this process. So this represents a chylomicron particle, and you have both ApoC2 and LPL on the surface. And LPL binds quite readily to lipoprotein or to lipids. But what happens is, as it undergoes lipolysis, you have buildup of surface tension, you have a buildup of free fatty acids and triglycerides, and it causes LPL to dissociate. So ApoC2 kind of tethers it to the surface, and this protein-protein interaction also probably activates LPL. But eventually, the surface tension builds up so high that both ApoC2 and LPL are displaced, and you end up with a remnant. If it hangs on longer, then you have complete lipolysis and you generate LDL. But most of the time, you don't go all the way down to cascade. Most of those particles are removed before they undergo complete lipolysis. Now, this is a slide probably is very familiar to many of you, and it relates to the pathogenesis of atherosclerosis. But the difference you might notice is rather than LDL, we have triglyceride-rich lipoproteins. So the very large particles are too large to enter, and they probably don't cause atherosclerosis. But when you have partially lipolyzed particles, they're about 20 to 30 nanometers in size, and LDL is about 20 nanometers in size. So these particles can get in, and then they can go into macrophage and generate macrophage foam cells, and then these initiate inflammation and cytokines. But what is different here than LDL is that unlike LDL is not taken up very readily by macrophages, where VLDL and remnants are very actively taken up by macrophages. So it actually contributes probably more to macrophage foam formation than LDL. And it's not clear why, but triglyceride-rich particles are much more pro-inflammatory. And one hypothesis is that the fatty acids that are generated are probably cytotoxic and damage cells. But this is very similar to how LDL, but except for the fact that it's probably more stimulates inflammation than LDL does itself. Now, I'm going to talk more about the kind of epidemiology of high triglycerides in a few moments, but this is an important slide that I wasn't really completely aware of. That I was always skeptical about this idea that these remnants were important, because when you look at the molar count of the remnants under normal lipid individuals, it's quite small. But when you get triglycerides above 200-300 mg per deciliter, you have quite a significant fraction of your cholesterol on the remnant particles. And you'll see in a second that on a per particle basis, cholesterol in remnants is probably twice as bad as LDL. So when individuals with triglycerides above 200-300 mg per deciliter, they're probably at an equal risk from cholesterol on their remnants than they are cholesterol on the LDL. And statins don't really lower remnants. So these are the people we want to treat if we can go further to further reduce cardiovascular disease. So in terms of what happens when people have high triglycerides, so as I mentioned, like many of you here in the audience, I work in a clinical laboratory, and we frequently see specimens like this. And it's usually, most of the time, it's patients that are on TPN who got intralipid, and it causes a big problem because it interferes with our measurement. But occasionally, it's actually patients that have genetic disorders. And it's very turbid because turbidity goes up by the cube of the diameter of the particle. So anything about VLDL and larger will reflect light. So this could either be VLDL or chylomicrons. And the way you test the difference, you just simply put the test tube in the refrigerator. Chylomicrons have a density less than water, so they float to the top, whereas this patient has very little chylos, and most of the turbidity is due to VLDL, which does not float. So what people are more familiar with probably is familiar hypercholesterolemia, where you have xanthomas, usually on your elbows and knees. They tend to last a long time. So patients with high triglycerides, they have what are called the eruptive xanthomas. They tend to be transient. They don't last a few weeks. They tend to be more diffuse. They're kind of yellowish in appearance, sometimes itchy. And what they are due to is infiltration of infiltration of macrophages in the dermis. And these macrophages are full of foam cells because they rarely take up this VLDL. And if you actively treat these patients, these xanthomas disappear very quickly. And if you do an eye exam, it looks like they have milk going through their blood vessels. It just looks like intralipid. And you can see that in contrast to a patient with a normal retinal exam. Now, the main problem these patients get that have these monogenetic disorders of very high triglycerides is pancreatitis. A few years ago, we had a young man who turned 21. He went out and celebrated and had, I think, 2 beers and pizza, and he had developed rip-roaring pancreatitis, high triglycerides, and he died. And pancreatitis is a potentially very dangerous disease. Many of these patients with the genetic disorders, they have pancreatitis every 2 months. So it's like Russian roulette. You have about 5% mortality with each episode of pancreatitis. So these patients oftentimes die of pancreatitis. In the U.S., there's about 300,000 admissions in the U.S. for pancreatitis. About 10% are due to high triglycerides. Most of those are not rare genetic disorders. You'll see that there's about 40 genes that regulate triglycerides. So if you're sort of on the edge and you take Accutane, and many times these are drug-induced, those are people who will reach levels that they can get pancreatitis. As I said, it's about 5% morbidity. There's limited treatment options. And the mechanism is not clear. The triglycerides, the highest I've seen is 10,000. And it becomes very viscous, so you might have actually ischemia reperfusion problems. Also, that you might have a two-hit hypothesis. You have pancreatic lipase, which is a secretory enzyme that helps. It's in the lumen intestine. It gets released into the tissue bed, and it starts to hydrolyze the circulating triglycerides and generate local high concentrations of fatty acids, which are detergents, and then it releases all the proteases. So that is thought to be the mechanism for the pancreatitis. Now, these are the list of the common causes of monogenic hypertriglyceridemia. Of course, if you miss the enzyme, you have that. And then what I'll talk about today, which is less common, is if you have a defect in ApoC2 itself. But I mentioned earlier that in the last 5 years, we now realize there's about 30 to 40 genes, they're all listed here, that modulate triglycerides. Most of these have a very modest effect. 5-10%. That's why we didn't notice them. And they're all discovered by Mendelian randomization and GWAS studies. So the typical person with triglycerides about 300 mg per deciliter, they have normal C2 and normal LPL, but they have a mixture, usually about 8 to 10 bad alleles in aggregate that accounts for their higher triglycerides. And this is shown here. About 1/3 of the U.S. population has triglycerides that are probably too high. Very rare individuals, less than 1% have triglycerides above 1,000. Those are the people that get pancreatitis spontaneously. But you have a large part of the population that has about 8 to 10 alleles out of the 40 that in aggregate give them high triglycerides. And on occasion, those people, when they go out and eat pizza or go on drugs that induce triglycerides, they can also develop pancreatitis. Now, this is a kind of a famous study looking at ApoC3, and I'll talk more about ApoC3 later on, but ApoC3 is kind of yin and yang of ApoC2. So ApoC3 inhibits LPL, and was first observed in the Amish. They have loss of function of ApoC3, they have low triglycerides, and they don't really have a great diet, but they're protected against cardiovascular disease. And this was now shown in many studies. And it's been shown now that almost all the genes that regulate triglycerides seem to not only modulate the triglyceride level, but also alter your risk for cardiovascular disease. So when I started in 1990, the Europeans were always believed in triglycerides, but here in the U.S. we thought the triglycerides weren't important, and that's completely changed in the last 5 years. Now we realize that remnants in triglycerides are pro-atherogenic or lead a causal way to atherosclerosis. Now, part of the reason there was skepticism in the U.S. is that fibrates have been around for a long time, and fibrates will lower triglyceride, they act as PPAR agonists, and the CORD study, for example, did not show benefit in reducing cardiovascular events. But those studies weren't really designed to ask the question that people with high triglycerides. And if you do post hoc analysis, and this was a study done by Mike Davis, and look for those individuals who had TGs above 200, they do show benefit from fibrates. And so there's now some prospective clinical trials that are trying to answer that question. Now this shows the CHD event reduction from triglyceride-rich lipoproteins versus LDL. And you can see that the strength association is stronger actually with triglycerides, which is kind of a surrogate for cholesterol on these remnants. And for every 1 millimolar decrease of LDL, you have about a 20% reduction in events. But for that same reduction in triglycerides, you have about a 50% reduction in events. And other studies have shown that the cholesterol on these remnants are about twice as pro-atherogenic in terms of this odds ratio than LDL-C. So what can we do about these patients with high triglycerides? So unlike the situation with LDL cholesterol, it's actually very hard to, I think the lifestyle modifications are beneficial for other reasons besides lowering LDL, but it's actually very hard to change your LDL cholesterol by diet or by exercise. But you can actually have a profound effect on triglycerides, and that's in part because exercise, for example, induces lipoprotein lipase, but only does it for about 24 to 48 hours. So you have to always be exercising to get that benefit. But lifestyle modifications are very important in high triglycerides, and diet is very important. So you have a bigger chance to modify that compared to LDL cholesterol. Now, the other thing that's also more important in triglycerides than LDL-C, that there's well-known secondary causes of high triglycerides. So the best example is hypothyroidism. And I'm always surprised how when someone comes in with high triglycerides, you need to check their TSH, and many times it's not done. And also drugs. A lot of drugs cause hypertriglyceridemia, so you need to treat the secondary causes. And as I said, that we have drugs such as fibrates, fish oils, and niacin that will lower triglycerides. Statins don't really lower triglycerides, but if someone's at risk, you need to always put them on a statin because lowering LDL is still important. And there's now some emerging therapies. So the first actually gene therapy approved ever was in Europe. It was for lipoprotein lipase deficiency about 4 or 5 years ago. And it was an adeno-associated virus expressing LPL in tissue. And it worked, however, it only worked for about 2-3 months. And then the gene expression stopped. And so this, I think, is no longer available. But LPL gene therapy may be resurrected. We have a project with Shire where we're trying to develop recombinant lipoprotein lipase as a potential therapy. There's some very exciting work with antisense oligonucleotides and also monoclonal antibodies that actually block these proteins that inhibit lipolysis. So, for example, ApoC3. And it's quite interesting that when you block these inhibitors and improve triglycerides, you also improve diabetes. And so ApoC3 antisense actually lowers hemoglobin A1C, and I'll talk about why that may happen later on in the talk. There are drugs called MTP inhibitors that are approved. They're mostly used for FH, but this is a protein that packages trilomicrons in with VLDL or ApoB. But the drugs are hard to tolerate and not widely used except for FH. There was great interest in DGAT inhibitors. This is the enzyme that esterifies, so the triglycerides are hydrolyzed in the lumen of the gut and then are re-esterified in the intestine and packaged. And so DGAT inhibitors are to block that re-esterification, but they cause fat malabsorption, and those drugs have largely been abandoned. And I'll talk about ApoC2. So ApoC2 is a very small protein. It's about 77 amino acids. And the most important part of the protein is the very C terminus. That's the part that activates lipoprotein lipase. So as I mentioned, during our patient rounds, we had a patient with ApoC2 deficiency, and he had a very bad case, and he would have pancreatitis about every 2 months. And he had his pancreas, he developed chronic pancreatitis and developed diabetes. So if you don't die of acute pancreatitis, you burn out your pancreas. So we thought, well, I thought at the time, so let's just make ApoC2. We can just synthesize it because I was making peptides. But what I didn't know at the time, let me jump ahead. Well, I'll talk about why we didn't do that. But so this is the structure of ApoC2. It has basically 3 helices. This is the part that activates LPL. This helix is very small. It's mostly a random coil. And this is a helix. So this is an alpha helix, and it's called a type A amphipathic helix. So all these residues in yellow and orange, they're very hydrophobic. And so this is what allows it to bind the lipoproteins, and all the charged residues on the other face. This is more of a globular type helix that you would find in the internal helix of a protein. And it's thought to be involved in a protein-protein interaction, and this will not bind to lipoproteins. So this peptide, even though this is the part that activates LPL, it doesn't work by itself. And we recently collected all the mutations. There's about 25 reported mutations in ApoC2. Only 1 or 2 that are common, and it actually is probably more common in African populations than in Caucasian. And it's about 1 in 100,000 is the homozygous frequency ApoC2 deficiency. So this is the NMR structure. This is the part of the protein that activates LPL. It's a globular type helix. This is mostly a random coil, sometimes called the second helix, and this is the part that allows it to bind to lipoproteins. Now, as I said, we thought we could just synthesize this peptide. It doesn't seem to have significant glycosylation, but what I didn't realize at the time is that even though peptide synthesis is quite efficient, each coupling is usually 90 to 95% efficient, but by the time you get about 20, 30, 40 residues, 95% over that length, you basically have no peptide left. So it's very hard to make a peptide, particularly the quantities you need, as a therapeutic when you get above 30 or 40 amino acids. So what we thought we would do instead is we took this last piece, which is about 22 amino acids, and instead of using the rest of the C2, we used a short helix of 18 amino acids that I've used for other projects that has actually been used in therapeutic peptides, and we linked it with a proline. So the total length was, I think, around 40 amino acids, so it was right on the edge what was doable in terms of making a synthetic peptide. These are the residues that are important in activation, so we made an active version by changing those residues to alanine. And we did this assay where we had our ApoC2 deficient patient. So normal individual, and I'll show you some examples later, that have high triglycerides, when you add LPL is almost all bound to the endothelial cells, so there's very little circulating LPL. So if you take plasma with a lot of triglycerides, you add the enzyme, it will undergo hydrolysis and you generate free fatty acids. And this is how you make the diagnosis of ApoC2 deficiency. If you have an LPL deficient patient and you add exogenous LPL, you will hydrolyze it. But if you have C2 deficiency, you're not missing the enzyme, you're missing the ApoC2, so nothing happens. But if you add LPL plus our peptide, we get good hydrolysis. If you add the inactive peptide, nothing happens. And then if you add ApoC2, you get lipolysis. So it appeared based on this study that our peptide, even though it's about half the length of the full-length protein, seemed to be equal potent to the full-length protein. So we actually examined carefully the potency of our peptide. So this is the last helix. It's inactive. It doesn't work because it doesn't bind lipoproteins. This is a truncated piece of the C2 protein that has that random coil region, but does not have the one that binds it. It works a little bit. This is the full-length protein, and this is our peptide. And you can see that if you look at the graph below, this is the same graph, but it's plotted with a log concentration. You can see that the peptide was actually has a lower Km, a higher potency, and it also increased the Vmax. So the peptide actually seemed to be superior to the full-length protein and activating LPL. So we wanted to make an animal model of ApoC2 deficiency. And this mouse was never made before, and I think for the same reason that the LPL knockout mice. So humans survive without LPL, but mice do not survive. They actually die shortly after birth. And we used zinc fingers. We just bought some commercial zinc fingers, and they happened to be right at the signal peptide cleavage site. So we had a lot of 1 or 2 base pair deletions with frame shifts. Those mice in heterozygous state survived. They had modest increased triglycerides, but the homozygous mice did not survive. But we had one mouse that was missing 9 nucleotides. He was missing 3 amino acids. As I said, this was fortuitous because this was at the signal peptide cleavage site. So the signal peptide was not cleaved. And what happens is they make C2, but it's larger in size, and it's been well described for other proteins that if you have a retained signal peptide, which is very hydrophobic, it is mislocalized. Either it doesn't get secreted, or in this case, it does not bind to particles with triglycerides. And so wild-type C2 binds both chylomicrons and HDL, but the mutant only bond to HDL, and this is where all the triglycerides are. So these mice had high triglycerides, particularly in the homozygous state, and they look like ApoC2 deficiency. So it's not a perfect model. We're now using CRISPR-Cas, and we're making conditional C2 knockout mice, and hopefully we'll have a better model for C2 deficiency. But this is the mice that we first tested, and this is my favorite slide I'm going to show you today, because it's quite dramatic. In fact, you don't need to measure anything. You just look at the sample. So these mice have very turbid plasma, and 15 minutes after giving the peptide, whether you give it IP, sub-Q, IM, doesn't matter how you deliver it, it just hydrolyzes all the triglycerides very quickly. And what was further remarkable, these small peptides usually have very short half-lives, just hours, but it actually kept it down for 2-3 days, and I'll explain why that happened. So we had a very remarkable effect in reducing triglycerides. It also lowered cholesterol, but those particles are mostly triglyceride, not cholesterol, so it doesn't have as a big effect on cholesterol because those are mostly triglyceride-rich particles. The other thing that was a little bit unanticipated is that the fatty acids actually went down, and as I said, because if you hydrolyze the particles when it's bound in endothelial cells, those fatty acids are sucked into tissues with CD36 or other mechanisms. So it was only until you overwhelm the system did they spill over into the plasma. So actually, fatty acids went down, and we think what was happening is we were sending the energy into adipocytes, and most of the plasma-free fatty acids is from intracellular lipolysis and released from adipose tissue. And so we were sending fat into adipose, and it turns off intracellular lipolysis. So it did not even come back after about 2-3 days.
Did the mice gain weight?
Yeah, I'm going to show you that. Yes, it did. Yeah, yeah. So, so as I mentioned, that the peptide, we made a fluorescent version of the peptide, and I was surprised how long it lasted. And this is just a gel filtration profile where we add the fluorescent peptide to plasma. If you add excess peptide, you get some unbound free peptide, but most of the peptide is either bound to HDL, LDL, or VLDL. But what normally happens is the C2 goes back and forth. So after the VLDL is consumed, it goes back. So this is remarkable because you just add LPL in the test tube, and then the VLDL gets hydrolyzed, surface tension goes up, and the fluorescent peptide moved over to HDL. So we think the long half-life of the peptide is that it was not free, it was bound to lipoproteins. And lipoproteins usually last 2 or 3 days. And this peptides, like the native protein, was probably going back and forth. And this is why we think we had that long half-life. So someone asked the question about obesity, but it wasn't quite, I think, the direction that you thought. So one of the things that we did is we looked at the mice on a normal child diet, and we found that the C2 knockout mice actually gained weight. So initially I thought, well, I thought too, like you did, that that the mice, that the knockout mice would be protected. But it turns out that even though C2 is important in adipocyte uptake, adipocytes are clever, and people have made LPL knockout. Ira Goldberg made a LPL knockout mice in the adipocytes, and they still get the energy in. So it was just the opposite I expected. Actually, the C2 knockout mice became obese. They had increased visceral fat, decreased skeletal muscle mass. They did not have a change in their fasting glucose, but fasting insulin went up, so they became insulin resistant. And we found this was true for both the heterozygote and homozygous mice. So I don't fully understand this, but as I said earlier, that if you antagonize ApoC3, it improves glucose control. And part of the explanation may be that the fat has to go somewhere. So normally when the fat goes to the skeletal muscle, you burn it. You also induce mitochondria. And if it doesn't go there, it ends up going back to the liver. So these mice actually develop fatty liver disease. So we think we might have got insulin resistance from fatty liver disease. And the fat still goes, the adipocytes don't seem to completely depend upon lipolysis to get energy. So, so we took OB mice. We did a glucose clamp study. And we treated OB mice, which you probably all know have a defect in leptin. And we were able to improve glucose control. So we think that the peptide, even though we're still mostly interested in triglycerides, may have an impact on glucose metabolism as well. So when I showed you that first study where we tested the peptide in plasma, we also tested normal plasma, and we had an unexpected finding shown here that I say about 5% of the population looks like they're ApoC2 deficient. So for example, this patient was 207. This is the free fatty acids with nothing. Adding LPL to that sample, nothing happened. So this is what you see with C2 deficiency, but we find about 5% of the population seem resistant. If you had LPL plus our peptide, we got lipolysis. And we find in general, almost every sample had more lipolysis with C2, which was a surprise to us because C2 is thought to be autosomal recessive disease, that you have enough C2 to go around. But it turns out that you have all these inhibitors, so it's probably the ratio of the inhibitory proteins to activator proteins, and I'll explain that later on. So it turns out that having more C2 may be useful for not only C2 deficiency, but maybe for other causes of high triglycerides. So we looked at that first in the ApoE knockout mice. They're missing ApoE, which is the ligand for removal. They have LPL and C2, and indeed, the C2 peptide lowered triglycerides in these mice. It decreased their VLDL, also lowered their cholesterol. And then I thought that, well, if you ever need more C2, when I stopped at the airport, I was starving, and I had a Big Mac. So whenever you have a Big Mac is when you need the C2. And it's actually been shown that a C2 in the intestine undergoes tremendous upregulation after a high-fat meal. It's about a 6-fold increase. So I thought, well, maybe this is when you really, so in the fasting state, maybe you don't need a lot of C2. But when you challenge the system, so what we did is we challenged the mice, we gavaged them with vegetable oil, and like humans, you can see their triglycerides goes up, it peaks about 2-3 hours, and by 4 to 6 hours, it starts to come back down. And if you give them the peptide, it basically completely blocks the postprandial rise. And it was due to hypercatabolism. And we did an agarose gel electrophoresis. So chylomicrons are too big, it gets stuck in the well, so these are chylos. And you can see the chylos have largely disappeared. The LDL and VLDL are gone. HDL, which migrates the fastest, was largely unaffected. And clinically, this is very important in diabetes. This is a diabetic mouse, the DB enos knockout mice. Almost all diabetics have increased postprandial lipids, and it doesn't come down. In fact, almost all day they have TGs are elevated because it doesn't come down. So we challenge these mice. Their TG goes up even higher than the wild-type mice, and again, the peptide completely blunts this increase. So we're thinking that you need your insulin right before your meal, and maybe combining C2 peptide with insulin might help with the postprandial rise. And diabetics die of glucose. They die of their lipids. So this could be very important, perhaps, in diabetes. So to look at this more carefully, I didn't think it was likely that the C2 was blocking absorption, but we bypassed absorption. We actually infused intralipid into the mice. And this is an example where you don't really need to do anything. You just look at the sample. So this is the control mice got intralipid, and these are mice that received the peptide. And again, so it was probably affecting the catabolism, not absorption of the lipid. And this is just shown here. In this case, we overwhelm the system, and free fatty acids actually go up because it saturates the tissue uptake. So then, as I said, we were suspicious that the whole story was maybe not just LPL, and that ApoC3 may be involved. So ApoC3, in addition to antagonizing LPL, does other beneficial things, removing lipids. So what we did is we gavaged mice with olive oil, and then we added LPL inhibitor. And if you block lipolysis, you now get TGs 4,000. If you give the peptide, it blocks it, as you saw earlier. But even in the presence of an LPL inhibitor, the peptide had a partial effect. So we think there was both an LPL-dependent and LPL-independent effect. So what could this LPL-independent effect be related to? So we thought this was due to ApoC3. So ApoC3 does inhibit LPL. So if you add ApoC3 to the test tube, it blocks lipolysis. And if you then add increasing doses of the peptide, you can overcome that inhibition. So this is human plasma was spiked with ApoC3. These are C3 transgenic mice, where we measure in vitro lipolysis, increasing peptide, we can overcome inhibition. And then this is why we did the experiment. So ApoC3, in addition to inhibiting LPL, also blocks hepatic uptake by proteoglycans or receptors. So this is intralipid that we add a fluorescent tag, and we then add ApoE, which is a ligand for uptake, and this is binding to the HDL receptor. We do flow cytometry, so increasing doses of ApoE enhance uptake, and then at the maximum dose of ApoE, ApoC3 antagonizes. So we think that ApoC3 probably has multiple effects, not only inhibiting LPL, but also interfering with clearance in the hepatic uptake. And given the mice, the peptide also lowered triglycerides quite dramatically, not as much as ApoC2 deficient, and it took much longer. As you saw earlier, about 15 to 30 minutes is in the nadir for the C2. It takes about 6 hours in case of the C3 deficient mice. So what is the potential mechanism? So it turns out C3, C2, and C1 all compete for the binding. C3 loosely binds to lipoproteins. And it's been shown by others that you can actually displace ApoC3 quite readily. So this is an SDS page gel. It's a little hard to see here, but this is C1, C2, C3. This is ApoA2 and ApoA1. And this is our peptide. So adding increasing concentrations of the peptide displaces all the APOs, including ApoC2. But in case of ApoC2, you're replacing it with a peptide that still activates LPL. So we quantified it here. And you can see that we decrease C3, decrease C2, and decrease C1. So the C3 is very small, so what happens is it gets filtered out by the kidney. So we're lowering C3 and activating C2. And we confirmed that. We developed a multi-TOF mass spec assay. We did in the C3 transgenic mice that were treated with the peptide. And C3 is very complicated. There's all these glycoforms. But we looked at all the different glycoforms of all these species. And both the human and mouse C3 were decreased about 40 to 50%. ApoC1 was also decreased, and mouse C2 to a small degree. So we think the peptide has both an agonist effect in regard to C2 and an antagonist against C3. So about 2 years ago now, we patented the peptide, and we licensed it to a company called Corvidia in Chicago. And Corvidia was started by a friend of mine named Mike Davison. Mike is a cardiologist at University of Chicago. And he started this company. He has a monoclonal antibody against IL-6 for patients on dialysis and licensed the peptide. And it's kind of a virtual company. There's only 3 or 4 people shown here, but they all have lots of experience in making drugs. And so they kind of helped me with the next phase I'm about to show you. So one of the problems with the peptide I just showed you is that that first helix is artificial helix and potentially maybe immunogenic. So we wanted to try to make a peptide that be more native. So what we did is we took this activation domain and we took this piece here, which you'll see is largely a random coil, but it potentially can form a nice amphipathic helix and bind lipid if we make a few changes. So we made it truncated where we used the native sequence and made a few changes and excluded this part of the peptide. So we made a whole series of peptides. I won't go into great detail, but this turned out to be the best one. This is the helix we're trying to replace, the artificial helix. You can see has a hydrophobic moment of 9, which talks about the polarity of the different proteins. This has a 6. Proline is very disruptive. It disrupts the helix, has this negative residue in the wrong place. So we took these residues and kind of changed them. And after doing so, we came up with this helix, which actually was a better lipid binding affinity than the 18A helix. So it's based on the native sequence, but we have 6 changes. So we're hoping this will reduce the potential immunogenicity of this peptide. And this peptide is equipotent to the original 18A-C2 peptide. So this is kind of new data, and I just have a few more slides, where we're doing a molecular dynamics simulation to try to understand how the peptide works. We did this on the Anton 2 supercomputer. And this is a kind of a trial layer where we have triglycerides. So one hypothesis is that this helix dips down and drags TG to the surface, because that's where the TG is a substrate, but it's actually in the core. And so we did some simulations, and we cannot really show that compared to an active peptide that it increased TG on the surface. I'm going to skip this to save time. We recently built a structural model of LPL. It's not crystallized, but there's homologous protein that is crystallized. So we built this model, and it's known to be active as a dimer. So this is the model of our peptide. And we did some cross-linking studies, and we found that the LPL, or the C2, actually binds to the LPL. The active site is here. This is a loop that shields the active site, so we think it actually moves the loop out of the way. So we're now trying to design our peptides to look at those particular residues. And then lastly, just 2 more slides. We have now done a lot of studies. We're starting to do studies in monkeys. Everything we did in mice worked wonderful. We didn't really have to do a lot of work with the formulation. Turns out monkeys are more challenging, and I think we have more work to do in terms of getting good sub-Q delivery. We're not getting the same level of sub-Q with IV, so we have not, unlike the mice, where we can lower triglycerides sub-Q, we can only get it to work IV on the monkeys. So we're doing a couple tricks to improve the bioavailability. But the half-life in monkeys is very long. These are small monkeys, so when we scale them up, it should have a half-life that it should be 3 or 4 days. It should be once-a-week drug, hopefully, in humans. And the sub-Q is not working yet in the monkeys, but the intravenous is lowering triglycerides by about 50% in the monkeys. So in summary, as you saw, triglyceride-rich lipoproteins can cause pancreatitis, but they are probably causally related to cardiovascular disease as well. A bihelical peptide containing a helix that anchors it to lipoprotein and one that activates LPL may be beneficial, but it not only activates C2 but antagonizes C3, unexpected, and it seems to blunt the postprandial rise in remnants, which may be very important, particularly in diabetics. And we're hoping to at some point identify our lead peptide and do toxicology so we can do a phase 1 study in the next year or so at the NIH. These are the people in my lab. Aki and Toshi are husband and wife, and they went back to Japan, and that's their new baby, Maki. And then Dennis and Lita and Anya kind of worked on the Delta 6 PV part of the project. So I would be very happy to, and these are my friends at Corvidia. So I'd be very happy to answer any questions. Hi.
Hi. I missed the first part of your talk, but the peptide you showed that it has 35 hours of half-life?
Yes.
Yeah, so I missed it, in the beginning. What's the difference? Because, the other kind of amphipathic helices, as my understanding, if you give IV, the L peptide
Right,
is
yes.
only like an hour.
Right.
So what is the difference between your peptide with other amphipathic peptides?
Yeah. So, well, as I mentioned, that it's even worse than that. That usually peptides, they're small. They get filtered out by the kidney, and so that's the challenge in using peptides as therapies. And so I think, as you saw, I don't know what part you missed, but a fluorescent version of peptide, it binds lipoproteins very well. And it may be that the lipid binding affinity is higher, and so there's probably an equilibrium between the bound and the free state. So in case of the L4F peptide that you worked on in Jerry Segrist and Anthony Mariah, that probably spends more time in a free state and probably gets filtered out more quickly. So it probably has to do with the relative lipid affinity, I'm guessing. But yeah, I'm not quite sure. We do have some evidence also that the peptide seems to bind to endothelial cells. So it may be not only binding to circulating lipoproteins, it may be binding to the tissue beds where the lipolysis also occurs, which would be good. But yeah, I'm still surprised how long it lasts. But yeah.
When you presented President Clinton's case, you said that he was a runner. But I noticed.
Well, he used to run to McDonald's, so he just like me.
I noticed that his HDL was only 45.
Right.
Do you have an explanation for that?
Well, well, so exercise will raise HDL, but you have to be a serious runner. So I don't think he was a serious runner. You have to do, you have to run like 4 or 5 hours a week. You have to do a lot of exercise to raise HDL. Yeah, so casual running will not raise your HDL. Yeah, but who knows? Maybe his HDL was even far less, and it went up.
So 2 questions. One, if you give people a drug to activate LPL, won't it make them fat, and won't they refuse to take it? And 2, what is the role of dietary carbohydrate in all of this?
Yeah. So we still recommend our patients with LPL deficiency to have a low-fat diet, but what happens is sometimes they switch and just eat carbohydrates. So carbohydrates turn into triglycerides. So yeah, one has to be very careful. That I think actually probably they're better off having a high protein. So yeah, we had this woman who had LPL deficiency, and she lived on Dr. Pepper, not Diet Dart, but Dr. Pepper, and she had terrible triglycerides. And as soon as she got off that Dr. Pepper, I think it was the fructose and the glucose, her TGs got better. So carbohydrates are important. In regard to your first question, I think that is a concern, but it's a balance. And I think if you eat too much fat, it has to go somewhere. So I think you cannot take such a drug and just eat the fat, because it has to go somewhere. But there is somewhat of a free lunch that if the fat goes to skeletal muscle, the fatty acids or PPAR agonists, it actually induces mitochondria. You actually will burn it. So hopefully, if you still have a good diet and you're sending the energy to skeletal muscle where it's supposed to go, you'll burn it up.
But isn't LPL dependent on insulin, and isn't insulin in turn dependent on carbohydrate?
Yes. Yeah. So it's very complicated. Yeah. I think one could actually maybe induce obesity with such a drug, but I think one has to control the diet and then use the drug. Yeah.
2 questions. One, on to the peptides.
Right.
On that. What logic did you use to get to modifying those 6 amino acids?
Yes.
Or was it just empirical? You went through everything and that's what you found?
Yeah, I went over it very quickly, but we basically looked at the helix, and one can predict its lipid binding affinity by calculating what's called the hydrophobic moment, and so we actually place them specifically to improve its lipid binding affinity. Yes.
Thank you. Second question was, has any work been done throughout life on ApoC2 levels? Does it change with age?
I'm not aware of any studies in humans, but there's been studies in mice and rats, and it changes quite dramatically during weaning. So that there's, like LPL itself, there's a marked increase ApoC2 and LPL when the mice are suckling, and then it goes down. So part of the reason the LPL knockout mice don't survive is that mouse milk, breast milk, is much more rich than humans in triglycerides, and they are much more heavily dependent upon fat metabolism. Human milk tends to be more carbohydrate. So these mice will survive as adults if you give them IP glucose every day. When they're born for 2 weeks, you can get them to survive. So yeah, so so I don't know in humans, but in mice there was induction of, and then the C2 goes down quite a bit after that period of time. So, and there is also some data that diet is that acute bolus of fat in mice induces C2 in the intestine.
Okay. No further questions?
So I'd like to thank everyone for coming.